Now, what you have done, you took your sample and run for single cell RNA-seq or scCITE-seq. And let's assume that you have done it using Genomics machine. And from that machine, the data that's generated there is really in FASTQ format. What do you want? I want to generate the analysis files from there. So those files could be in .h file or any of the matrix format. To generate those, what you have to do is at the very beginning, look for the next Genomics cloud analysis platform. So when you enter into the website, you'll see there's something called create project. So for example, you write it like let's assume we want this to be site data. So created the project. Now the question is that we need to upload the files, and then only we can use this cloud platform of Genomics for analysis. So this use the star-based methods, which is common in terms of bulk and single cell RNA-seq methods. You can use other tools also, but we are going to demonstrate this today. So let me go. Okay, what do we see? We can simply do browse files and select our files and just click open. It will start uploading. But these files are really big in size and we want to upload them cautiously without breaking the upload speed. So for that, they have certain instructions that if you're using Linux system, you can use the Linux command line to upload these files. You have Mac and Windows system. So as I have the Linux system, so what I do at the very beginning, just take this, follow this instruction. That's simple. Paste it. Let's download the tar file. This file will work as an interface to connect the Linux PC with the cloud platform to Genomics. So you have connected it. Now what they have said, we need to unpack the tar file. So we unpacked it. And the next first is to go and check within this folder. That's how we go within this directory and then we use it for our further processing. So we copy the directory. Let's see. So this is we are within the directory. So in the next phase, what we are going to do is that we want to upload the files for one whole project within one folder. And to do so, what we will do, we'll follow certain command from their website. So what are the kind of websites command they have? So if you go in here and look for the tutorial, we'll say this is tutorial. They have the PDF tutorial also to upload it. Within here, we want to upload the entire folder of the FASTQ files for one specific study. And to do so, what we will do in here to upload the FASTQ files in a folder that begins with something. So we'll optimize it for ourselves. We go in here, paste this code, and then at the very beginning, you can see there's a project ID. So where do we get our project ID? So back in here, you can see the project ID is here. Copy it. Let's paste it. Then the next one is file name. So let's copy our file name. Let's assume our files are in this specific folder. So copy our folder in here and then click. So this, if you're doing it for the first time, they will ask for the access token, and you can find the access token in here. But either way, it's specifically asking to upload the file. So and it starts uploading, and the uploading speed you can easily see. Speed will be up. So now the easiest part, if you want to upload your FASTQ files, simply click on this. This is the way you can upload, one way, and another way is that just click on browse. Now if you want, okay, this is my folder. Is it fine? So we want this entire folder to be uploaded. So select, click. Then the option comes start uploading, and the upload will process. All the files will be uploaded one by one. This is as simple as it. Let me know if you have any confusion. Bye.